Good morning. My name is Liv Williamson. I'm from the University of Miami, and I'll be talking with you about our efforts to breed Diploria labyrinthiformis for stony coral tissue loss disease resistance. Since its outbreak in 2014 off the coast of Miami-Dade, stony coral tissue loss disease has devastated coral populations in Florida's coral reef and far beyond. This disease affects over 24 species and causes rapid infection and mortality, making it perhaps one of the most lethal and prevalent diseases that we've ever seen in corals. Now, despite dozens of studies documenting the impacts of this disease on our corals, so few of them have focused on how this disease might impact coral recruits and juveniles. And we thought this was a major gap because sexual propagation is really being scaled up right now, especially with stony coral tissue loss disease susceptible species. So earlier this year, we published a study that found that unfortunately several species of coral recruits are impacted by stony coral tissue loss disease in similar ways as their adult counterparts. And that, of course, led us to being interested in looking into some ways that these recruits might become more resistant to disease and finding some silver linings and ways that we can actually make productive studies out of this information. So that led us to making an observation. There are many, many colonies in the endemic zone that have somehow survived for years and years with high levels of mortality and infection all around them. So presumably many of these colonies might have some level of resistance to stony coral tissue loss disease, which makes them really interesting for restoration practitioners, both for use in asexual propagation and of course for use in sexual propagation as parents, because potentially part of this resistance might be genetically based or at least otherwise heritable. So going forward, I'm going to refer to these sorts of colonies that have survived in the endemic zone and are presumably somewhat resistant to stony coral tissue loss disease as endemic colonies. Now, as our friend Carly Kenkel told us yesterday, if we're going to be thinking about using these as parents for managed breeding, we can't only use them. We don't only want to be taking a small subset of corals that we somehow deem to be more stress tolerant than others and only using them to make a new generation. We want to capture as much genetic diversity as possible. And that leads me to the Florida Coral Rescue Project. So ahead of the disease front as it moved across Florida's coral reef, scientists led by the Department of Environmental Protection here in Florida actually removed hundreds or thousands of colonies of these susceptible species from Florida's coral reef and put them into zoos and aquariums to serve as a living gene bank. Now, the idea was that these colonies probably will never be returned to the reef. They had never been exposed to the disease, so they are presumably somewhat susceptible to it, but that they could be used as broodstock and that their offspring would eventually be put back onto the reef, so this genetic diversity would not be lost in Florida's population. So we wanted to take advantage of this great resource, and we thought maybe we can sort of accomplish both the goals of increasing genetic diversity in recruits while also making sure that those recruits have a little bit of disease resistance in them by attempting to breed together these endemic colonies with these rescued colonies. And last year we had a chance to do this. So, on a fateful night in May 2021, at the Florida Aquarium, Diploria labyrinthiformis spawned, and these were the rescued colonies. On that exact same night, endemic colonies in Miami also spawned. And of course, in those respective institutions, we mixed together those gametes and made cohorts of larvae and recruits right there. But we also cryopreserved sperm in each of those locations. And the next day, that sperm was driven across the state and exchanged, so that the next night, when colonies spawned again, in both locations, fresh eggs could be mixed and fertilized with cryopreserved sperm from the other location. And in both cases, this created cohorts of offspring that had one endemic parent and one rescue parent. So going forward for the experiment I'll be talking about, each of these three sort of different parentage cohorts are included. On those same nights, another team of divers in Key Largo also collected spawn from endemic colonies on the reef and created offspring from these. And so that is a fourth batch of endemic parent colonies that is also used in the experiments I'll be discussing. So for each of these four batches of cohorts of recruits, they were reared for 5 months in their respective institutions, after which they were all transferred to the University of Miami for a month of sort of acclimatization in our systems before being exposed to this treatment. So then we put recruits from each of the 4 different cohorts into 8 different aquaria, 2 of which were treated as control aquaria and given water from a bath of apparently healthy coral colonies, and the other 6 were treated as sort of disease-exposed treatments. They were given water from a bath of coral colonies that showed active stony coral tissue loss disease lesions and tissue loss. The recruits were maintained in these treatments for 30 days, during which they were regularly monitored and photographed for survivorship, disease incidence, and lesion progression. So what did we find? Although unfortunately, some recruits from each cohort did develop lesions and eventually die from those, the recruits from the Florida Aquarium, which you'll remember have two rescue parents, two naive to disease parents, had significantly higher incidence of disease than any of the other cohorts, all of which had at least one, if not two, endemic parents. Lesions look very similar in recruits as they do in adults. They typically start on one edge of the colony from a focal or maybe multifocal lesion and progress their way across the colony, or the polyp in this case, to eventually result in completely denuded skeleton. It's really sad to see this on the little baby guys. So at the end of the 30-day exposure, we found that the Florida Aquarium recruits, which again have two rescue parents, had significantly lower probability of survival than any of the other cohorts, and particularly about half of the probability of survival of the cohort from Miami, two endemic parents, and the Miami times Florida Aquarium cohort, which was an endemic and a rescue parent. So this is pretty promising if we believe this to be true, right? Having one endemic parent gives you just as high a probability of surviving a 30-day dose of stony coral tissue loss disease as having two. That would be awesome if that were true, and it might be, but we need to caveat these results. So we have funding to be able to do genetic analyses on this cohort that is Miami times Florida Aquarium, but for logistical reasons, we haven't been able to do that yet. So I can't definitively tell you that those recruits are for sure, having one parent that's endemic and one parent that's rescue. We did do a no sperm control, of course, and we found less than 5% fertilization in that. So I'm pretty confident that these recruits are what I think they are, but I can't give you these results without giving you that caveat, and also looking at these results through the lens of assuming that maybe that cohort isn't actually mixed, and that perhaps these babies were fertilized by Miami sperm prior to being fertilized by cryopreserved Florida Aquarium sperm. So if we instead group that Miami and that Miami times Florida Aquarium cohort together and assume that they're one thing, our results hold pretty, true. We still see a much higher probability of survival, significantly higher probability of survival in this Miami cohort. These babies with two endemic parents seem to be doing a lot better than these two rescue parents. And you'll notice that the Key Largo cohort is somewhere in between. So this is really interesting to us. We also let these recruits all recover for a month after this experimental treatment. So we put them into quarantine tanks, and we're no longer, dripping them with active disease water, and kind of found a pattern that looked very similar to the survivorship that we saw during the exposure, whereby after a month of just hanging out and being allowed to recover, actually quite a few of them did continue to develop lesions and die, and we saw much higher mortality in those Florida Aquarium recruits that had survived that initial exposure. So I'm really encouraged by these initial results, but of course those caveats are really important to remember and make me just want to go in lots of different future directions to keep answering these questions more robustly. So first of all, genetic analyses are going to be totally essential for any managed breeding programs that are using endemic and rescue colonies together, especially for parents that are sort of the cryopreserved sperm side of things, so that we can make sure we verify parentage for all of these crosses that we're making, but also so that we can track if there are any specific parent colonies that are disproportionately contributing to recruits that survive these disease exposures. Similarly, we would really like to be able to cross fresh gametes, both eggs and sperm, whenever possible, because the cryosperm really does create a problem of scale. We can only freeze so many vials at once, we can only thaw so many vials at once. So unfortunately, right now where we are with that, we just can't create the number of larvae that we need on a restoration scale. And luckily with this, the Florida Aquarium actually now has colonies that are both rescue and endemic in their XT2 spawning systems. So we're hoping that in the future, those will spawn on the same nights so that we can make those fresh crosses basically in real time and alleviate some of those issues. At the same time, we're interested in doing reciprocal crosses with rescue and endemic parents, because studies out of the Indo-Pacific show us that the heritability of heat tolerance might actually be different if it's coming from your mom or your dad. So perhaps that's true with disease resistance as well, and that's worth looking into. There's also growing evidence that symbiont communities do impact a coral's susceptibility to disease. My colleague Carly Dennison gave a great talk earlier explaining that breviolum seems to be associated with higher disease incidence. And although I did sample all of these recruits before they went into our exposure and found that they only seemed to host Gerstinium trenchi, it's possible that in small quantities there were other symbionts present that could have somehow influenced their susceptibility. And then finally, of course, we want to think about breeding other species. There's so many species that are susceptible to this disease, sadly, and many of them are actively being propagated for restoration purposes. So we want to make sure we're not just sending them out there to be, skittledy, guinea pigs when we put them out onto the reef. So in conclusion, we think this is the first study to attempt to breed corals for disease resistance, and we're hoping that it might help to inform whether this is a viable management action going forward for this disease and others. Unfortunately, I did not find any cohorts that seemed completely resistant to disease. This may be because we gave them a pretty high dose that might not even be ecologically relevant, but I do think it really flags for me and underscores the urgency of making sure we consider recruits and juveniles when we're thinking about stony coral tissue loss disease impacts. We should not forget about them, because if the recruits we're putting out there are dying, you know, the natural or assisted population recovery is just not going to work. I am really encouraged by our results, and it seems that these endemic parents that have survived for a long time in the stony coral tissue loss disease endemic zone might be able to pass on some degree of that resistance to their offspring. So I certainly suggest that we prioritize these within managed breeding schemes. But again, as Carly Kinkel said, we cannot forget about the other colonies that also will make sure that we have very genetically diverse offspring and continue to use those as well. So in conclusion, I hope that we're all considering lots of different intervention strategies that we can test and use in coral restoration, and I hope that managed breeding might be one of those that can particularly help us in the face of this epidemic we're facing. Acknowledgements. Thanks so much to my sources of larvae and my funding sources, NOTAS3 and Florida DEP, and collaborating institutions and folks that have helped us out with these experiments. Thanks so much.
Great talk. We have time for questions. Yes.
Hey.
Lynn, I can't remember if you went over this at the beginning, but are the corals at black, are they all